Hi, everybody, and welcome to another installment of our Closer Look Open Journal Club in Biomechanics. My name is Matt Bursey, and I'm joined here today by the co-organizers of the Journal Club, Immobilization, Manuel Rausch, Adrian Buganza-Tepele, and Johannes Weckenmeyer. If you're new here, and this is your first time, welcome. The goal of the Journal Club really, whenever it was conceived, is to bring together researchers and students across the biomechanics community to learn about some recently published work from various experts. So along those lines, today we have the pleasure of hosting Professor Spencer Lake from Washington University in St. Louis. Spence received his PhD from UPenn prior to starting his postdoc at University of Minnesota, and then moving on to his current faculty position here at WashU. Spence is an associate professor in the Department of Mechanical Engineering Material Science. His lab is, I would say, generally focused on musculoskeletal and orthopedic biomechanics with various applications. And a lot of his work has yielded contributions in the field of tendon mechanics, as well as injury in the elbow joint, which I think we'll hear about today. So with that, yeah, we're excited to hear about some of the ongoing work, looking at the role of collagen crosslinks with the title of his talk shown there on the slide. So with that, I'll yield the floor and excited to hear from you, Spence.
Excellent. Thanks for the introduction, Matt. And yeah, the title, I wouldn't normally have a title this long, but this was the title of the paper, so I left it as is. But this is the work that I was invited to talk about. I'm really excited for this opportunity. So my lab is the Musculoskeletal Soft Tissue Lab. Our kind of tagline is multi-scale structure function relationships with musculoskeletal soft tissues and joints. We have three main projects that we're working on right now. Multi-scale tendon mechanics. We do a lot of polarization imaging to look at tissue organization and microstructure organization in tendons and ligaments and other tissues. And then we have a third major project, which is post-traumatic elbow contracture, which is what I'll be talking about today, the papers on that topic. So what is post-traumatic elbow contracture? Of course, you know how an elbow works. It's a hinge joint, but also has a rotating pivot joint. So there's two main motions associated with that we need for full function: flexion and extension, and pronation, supination. Typical range of motion ranges in a human are shown here, and kind of the standard for a good functional range is about 100 degrees of motion in each of those directions. The elbow is commonly injured. Dislocations are quite common, even though this is an inherently stable joint. And then complex fractures, often with dislocations, are also fairly common. And a challenging outcome from these kinds of injuries is a joint contracture that develops in the healing and recovery phase. And this is very unpredictable. It's hard to know who's going to lose range of motion, but it can be quite severe, and it's really challenging to prevent and to treat once it's developed. So there are ways that surgeons are treating, as well as physical therapists. So physical therapy with splinting or manipulation of the joint can be helpful if it's done early. It's not always successful, and following unsuccessful efforts, then surgical treatment is one potential remedy. So about 12 to 15% of patients will require a surgical release of the capsule, where really it's a surgery to just cut away the anterior capsule on the front side of the joint. And this does usually improve range of motion, although a couple studies have said only about 50% of the lost motion is regained even through this fairly invasive approach. And then other options that are even more debilitating would be total elbow arthroplasty, so replacing the whole joint, or in severe cases, joint fusion. So not great options. So prevention and treatment strategies are really needed. And what I don't have here also is prediction strategies to understand who's going to develop contracture is also desperately needed. So I'll cover kind of some of the main objectives of our work in this area. One is just determine the etiology and progression of the fibrotic response. So in other words, what happens after an injury, like a dislocation to the elbow joint, and identify which tissues contribute to that response. So which tissues are, changing biologically, and how does that impact the long-term mechanics? And then what we're really interested in is trying to develop treatment strategies or prevention strategies to maintain range of motion of the joint, and then hopefully leverage results from our preclinical studies to inform clinical approaches to really have an impact on patients ultimately. So to undertake this work, we decided we really needed to use an animal model so we could really dig deeply into the biological response and changes that are occurring in tissues following an injury like this to the elbow. So when I started this project nearly 10 years ago, when I started at WashU, I partnered with an orthopedic surgeon, Lisa Gallitz, and she said, this is a real clinical challenge. Let's see if we can figure out ways to address it. And we decided to pursue an appropriate animal model. So we looked at a number of different species. We excluded some due to ethical or access concerns, but we looked at a number of criteria. So range of motion, both in flexion, extension, and pronation, supination. We were looking for animals that actually would pronate and supinate the joint in addition to just flexion extension. Looked at anatomical similarities, the functional use of the limb, and then just some feasibility considerations as well. And we did a number of different things to try and evaluate different options, and consulted with veterinarians and different things like that, and ultimately settled on the long Evans rat. And of course, rats are advantageous for a number of reasons. They're small and relatively cheap and easy to use, and there's quite a few assays developed. But I'll just make a comment about this specific breed. We found some really interesting studies from a number of years ago looking at joint motion in long Evans rats, even compared to other breeds of rats. And it turns out that these long Evans rats have more pronation supination motion. And there were studies comparing their motion during eating to human motion and actually found more similarities than you would expect. So it's kind of an interesting kind of fortuitous find in a couple of published papers on that topic. So in terms of how does that compare? So here's a micro CT of the rat elbow. And it's actually pretty similar. There are 3 distinct bones, like the human, although not all mammal species did have the 3 distinct bones. The joint articulations are fairly similar. One noticeable difference I'll point out is the olecranon is kind of the back end of your ulna. And in humans, it ends at the joint. And in rats, you can see on that far left image, it extends back past the joint, such that rats can't extend their elbow to 180 degrees, which actually 0 degrees of extension is that the way they refer to that. But besides that change, most other anatomical considerations were pretty similar. So we wanted to recreate an injury model in the rats that replicated what's seen clinically in humans. And so we targeted 2 tissues in particular, the anterior capsule, which is a joint capsule on the front side. There is a posterior capsule as well, but we target the anterior capsule and the lateral collateral ligament, which is this ligament outlined in red, because these are tissues that are typically torn in a dislocation. So if you dislocate your elbow, often you have damage to these 2 tissues. So in our injury model, we cut those, punctured the anterior capsule, and we transect the ligament. And these are a couple of images during surgery to give you an idea of that. So this is the rat limb. Panel B, you can kind of see the outline. Here it's labeled LUCL, but it's the lateral collateral ligament. So we can cut through that. And then panel C, you can see the exposed. It's a little harder to see, but you can kind of see where the anterior capsule sits right there on the front side of the joint. And we puncture that with a scalpel. And then panel E shows a full dislocation of the radial head. And then we close this back up. And following surgery, we immobilize the rats using some elastic net bandage and then a vet wrap bandage that we wrap around the limb. And I'll just mention that compared to nearly every other model we've ever seen, animal model of immobilization, this is the most kind of relevant clinically. Usually there's a pin that's placed internally or a wire that's wrapped around the bones within the joint. And we tried to replicate a more normal placement of the limb in a soft bandage. So then our typical time course, this varies a little bit in some studies, but most of what I'll be talking about today is 2 groups. So our control group, we allow what we call free mobilization, which is just a cage activity, unrestricted motion. And then our injured group is the injury at day 0, immobilized for 42 days, and then given 42 days additional of free mobilization. So we take them out of their bandages, they have cage activity for an additional 6 weeks. So like I said, those are kind of the 2 standard time points that I'll be talking about. And we did a lot of evaluation. We've done a lot of evaluation at 42 days, right when they come out of their bandages, and then 84 days after they've had that additional free mobilization period. Okay, so one of our first questions after having developed this model is, which periarticular tissues, which tissues in the joint really contribute to the contracture? And we focused on 3 initially. So looking at the capsule and the ligament, which of course are injured tissues. So how does the healing response of those tissues contribute to loss of motion? And then we also want to look at muscle changes and see if that was a long-term contributor. I added cartilage and bone here. I won't talk much about that today. We've started to look at changes in these tissues as well, not so much directly contributing to loss of motion directly, but changes that are occurring there that might have some signaling implications in the joint as well. So to highlight a few of the first things we did with this animal model. So we developed this model and showed that there is indeed long-term contracture. So this is a video kind of from above of a flexion extension elbow joint mechanical test using a rat limb. So we've potted the limb, both sides in a following dissection. We've only removed the skin, but the other tissues are all there. Let's see if I'll put it one more time. And so we can track flexion extension range of motion. We also did a study where we looked at pronation, supination motion. I'm not going to talk about that today, but we also can evaluate that mechanically and quantify those results. So we get a number of output parameters. I'll focus on total range of motion, which is just the range of that motion to certain fixed torque limits. And the first 4 bars here under IM, these are at different periods of immobilization, and then sacking the animal at the end of that immobilization period and looking at range of motion. So we see with more immobilization time, you get a decrease in range of motion, not terribly surprising. And then these, the pink and the red bar are after 21 days or 42 days of cage activity free mobilization. And we see that there is there's a slight improvement in range of motion, but a persistent decrease. Manuel.
Sorry for interrupting. If I missed it, I apologize. But I mean, your motor could impose any range of motion. How do you determine what is the limit of your range of motion? Sorry if I missed that.
Yeah, no problem. So at the other end of this is a rack and pinion gear. So at the other end of this rack is the load cell. So we're measuring forces and then just converting that to torque. So we did preliminary testing and identified some torque limits that kind of represent a reasonable limit at which the limb should be moving. And so we just impose that as a force control determinant. Okay, so this established that over time we do get a decrease in range of motion. It recovers slightly, but remains decreased compared to control. So then we started to look at which soft tissues are contributing to the contracture. So one thing we did was a sequential dissection mechanical testing. So in other words, we tested the limb like I just showed, and then we dissected away the capsule and retested, and then dissected, muscle and tendons, retested, dissected away the ligaments, and then finally dissected the capsule and did repeat testing. And so doing that sequentially, we could look at the relative contributions in terms of, here I've plotted in terms of extension lost, because this is a contracture inflection. So we're talking about lost ability to extend the limb. So after that 42-day time point, most of the contracture is really due to the capsule tissue, and then a little bit to muscle, and really not much due to ligaments, at least according to this set of mechanical tests. At 63 days, there's less extension loss, right? There was a little bit of gain in motion, and there's a shift now where about half the lost motion is due to the capsule tissue, and now about half is due to ligament cartilage, with basically no contribution due to the muscles in the joint. And then looking at 84 days, it's even a little further shift of kind of ligaments, other ligamentous tissues, capsule still contributing, but not as much as previously. And again, really no contribution from the muscle. So this kind of highlighted to us that the ligaments and the capsule are playing a role in terms of the mechanics of the range of motion loss. We wanted to confirm that muscle wasn't having a long-term effect. And so we did a whole additional study where we looked at changes to the muscle using histology and active and passive muscle testing. And I just, a little snapshot here is that we did see changes at 42 days. So after the mobilization time period, this is the blue bar here and the blue dots on the right. So we did see some changes early after that mobilization period. But once we remobilized the joints and they had full use of the limb for an additional 6 weeks, then all the mechanics and the histological assessment all returned to normal. So we saw no long-term contribution from the muscle. So our next step was, can we evaluate in more detail the anterior capsule and this LCL, this ligament, to really try and figure out what is causing the contribution to this joint deficiency. So we had already previously done, prior to the present study that I'm talking about, we had done a little bit of histological analysis of the capsule. So here's a toluidine blue stain and an H&E stain. And we work with a musculoskeletal pathologist who'd done some basic scoring. And we see an increase in fibrosis score and adhesions. We see increase in a little bit increase in proteoglycans and myofibroblasts and fibroblasts. And then increased thickness of the capsule. So we've done this, and so we had some sense of maybe a little bit what's happening, but not really much detailed look of changes that were occurring. And previous to this study I'm talking about today, we hadn't done anything to look at the lateral collateral ligament because it's a little trickier to access histologically when we're making sagittal cuts through the limbs. Okay, so what were some of the unknowns that we were trying to address in this study? So first few things would be, is there hypertrophy of these tissues? We saw in the histology that the capsule is a little bit thicker. Is there a mechanical consequence to that? And does the LCL also show hypertrophic changes? Is there an increase in tissue volume there? What about compositional changes? Quantity or density of the collagen? Ideally, we'd like to know other types of collagen. I'll mention that we didn't actually get to that until this point. And then just other compositional changes, increases in GAGs and other things. And then we got really interested in looking at changes to collagen crosslink. This was spurred in part by a presentation I gave at the World Congress of Biomechanics 4 years ago. And I had a question from Savio Wu in the audience, who many of you may know. And he asked me about crosslinking, and I said, we hadn't looked at that point. And he said, I think you should look at it. And he was right. And so we did start looking at that in this case. And then really, how does an understanding of these changes provide information to help us perhaps identify appropriate targets for treatments or prevention strategies? So I'm going to take a slight detour and talk more about the collagen crosslinks because I think it's a little more unique aspect of the protocol and maybe perhaps why this paper was selected for me to talk about. So I'm going to talk a few minutes about collagen crosslinks. Acknowledging, of course, that I'm not a biochemist and don't know this as in detail as our collaborator, but I'll give you kind of my perspective on it. So collagen molecules are stabilized by these intermolecular crosslinks. So you look at the top image of the schematic, you see a triple helix collagen molecule. And then, a lot of the chemistry that's involved in crosslinking, and I'll just highlight a couple things. So there's an enzyme called lysyl oxidase, or LOX. That's the little green shape pattern over here. And this converts these telepeptidyl, if that's the right way to say that, the residues at the end of the collagen molecules into aldehydes. And then those can then react with hydroxylysine or lysine on neighboring collagen molecules and form these covalent bonds. And initially, these form as a divalent bond that's been called an immature crosslink, and that's shown here in the kind of the green box. And then over time, with maturation of the tissue and of the collagen, these divalent crosslinks condense to form a mature crosslink that's a trivalent. And so a little bit different chemistry there, linking one of these triple helix and then two of these residues. Now, at the bottom of this schematic is something I'm not going to really talk about at all today, which is another type of crosslinking that I'm sure you're aware of, which would be like glycation crosslinks. We did not look at that in this study here. Certainly could be relevant in a number of scenarios, certainly in clinical conditions where there's dysregulated sugars and just in aging in general. But what we've looked at so far are really these enzymatic crosslinks highlighted above. So here's kind of spelling this out a little bit more detail. So we looked at enzymatic collagen crosslinks, and there's a really nice review paper by Gar in 2020 that I'm going to show a few images from. But it turns out there's a lot of different collagen crosslinks that are enzymatic. And so you can see that it becomes very quickly a very complex topic to try and undertake in terms of deciding which of these to look at and which might be important. In connective tissues, like that we've looked at, and in my background in tendon, there's a few that are more commonly evaluated and more commonly present. So we focused on those. So we looked at two immature crosslinks that have long names and perhaps equally long acronyms, but DHLNL, which is still a mouthful, and HLNL are the two that we've looked at that are these divalent immature. And then we looked at 2 mature crosslinks, hydroxylysylpyridinoline HP and lysylpyridinoline LP. So those are the ones I'll be focusing on today. And then, like I said, there are other kinds of collagen crosslinks. So advanced glycation end project ages-based crosslinks like pentosidyne is a common one, but there are actually a whole variety of these, some of which are not very well studied. But okay, so what has been looked at previously? Well, it turns out this topic was looked at many years ago, back in the 60s and 70s. And this was some of the work that Savio Wu was referring to when he asked me this question. So there was a study in '66 that looked at knee joints from dogs, rats, and then a couple of cadaveric human fingers, and looked at contracture. And their conclusion was that the contractures do more to collagen synthesis rather than cross-linking of collagen or older collagen. And through a couple of manipulation experiments where they induced cross-linking through dehydration and through formaldehyde, they determined that the effect of crosslinking was too small to be impactful. But I'll mention that the experimental approaches were pretty primitive. And so like they measured crosslinks by just heating up a bath with the tissues in the water and looking at what temperature the tissues shrunk. So anyway, I would say it was an interesting study, but perhaps limited a little bit by the availability of techniques. Although the same investigator a year later appeared to have changed his mind, because then he decided to look at reducing collagen crosslinking to see if that would prevent immobilization or prevent contracture in immobilized rat knees. So using these rats, they treated the animals with BAPN, beta aminopropyl nitrile, which is used to prevent crosslink formation. And they did see some improvements in joint stiffness with a 3-week BAPN treatment, but not with a 1-week dose. But this was an immobilization only model and did not directly measure crosslinks, just applied this drug to try and prevent that. And about 10 years later, Savio Wu's group had a couple of studies where they looked at immobilized rabbit knees and did actually look at measuring collagen crosslinks using chromatography. And they saw some increased amounts of crosslinks in those immature crosslinks I mentioned, as well as a desmosine crosslink, which is more commonly associated with elastin. And they tried, in the subsequent study, they used penicillamine to reduce collagen crosslink formation. And they did see a little bit of reduction of crosslink, but saw an increased amount of these crosslink precursors, which perhaps is a little unknown what the long-term effect would be then following a discontinuation of the drug, if then the amount of crosslinks would change as a result of that. So a very interesting study. The other limitation of this study is you can see the drawing on the right of the contracted rabbit joint, and they basically harvested all the tissue together and processed it. So it wasn't separated out in terms of which tissues were involved. But anyway, this laid some nice groundwork for investigations of this. But then really since 1977, there's been very, very little looking at collagen crosslinking in immobilization. So we sought to undertake this, and we thought, well, if we're going to do this, we need to be able to measure collagen crosslinks. And so a few years prior, I had wanted to look at crosslinks in tendon, and I talked to a friend of mine who was at WashU at the time. He's since moved to London, but Jan Bischke, I talked to him about doing chromatography to measure crosslinks using some protocols we'd found. And he said, yeah, sure, no problem, we can do that. So we submitted the proposal, we got the award, and we said, okay, we're ready to start measuring these crosslinks in tendon. And when he looked more carefully, he said, oh, actually, I can't do that with my system. You have to be able to measure fluorescence, and so I can't help you. So that was a kind of a dead end. So we started talking to other people. And we talked to Kristen Myers, who's at Columbia, and she had measured crosslinks in, I think, in the uterus or cervix, and had done some really nice work there. And together with her postdoc or grad student, I can't remember, Kyoko was grad student postdoc, but they had some really helpful conversations with us and kind of gave us some initial protocols. And they referred us to a cohort at Columbia at the Irving Institute that did biomarker analysis. And so we got in touch with them and were able to send some samples. And initially we had a great result. We sent some tendon samples and measured some crosslinks, and we were very excited and figured we'd sorted this out. So then when I came to do the study we're talking about now, we got back in touch and said, can we send you some elbow tissues from our rats? And she said, Well, actually, we can't do that anymore because the protocol is kind of a dirty protocol and it kind of messes up our system and you have to clean the whole thing. So we're not doing it anymore. So then we're back to the beginning. We're trying to figure out where to do this. So I had some conversations with Catherine Kuo, who's a friend who had also done some crosslink analysis in chick embryos, tendons. And she was in the process of moving from Tufts to Rochester and has subsequently moved to Maryland, but was kind of right in the process of moving, and so really couldn't help us there either. So we're thinking, what are we going to do now? And then I happened to talk to Dirk Humacher, who was at the time was the scientist at Cleveland Clinic, who's now at Mount Sinai. And I was talking about doing this, and he said, "Oh, I have a friend who does that. I should get in touch with him." So he got us in touch with Jürgen Brinkmann, who's at University of Lübeck in Germany, and we got in touch with him. And it turns out he's been a fantastic collaborator. We've been able to measure this. So you might wonder why I spent all this time on this slide. Well, one is because I had to just make the joke that I've established a crosslink network of people to be able to measure this. But really just to show that, you know, it's been 40 years since someone's really looked in depth at cross-linked tissues in Contracture. And I think maybe this highlights one of the reasons why is because the technique itself is not really that challenging, but having the appropriate equipment and the expertise to be able to run it, for us at least, has been quite challenging. So I'm happy to report we can now do that with our friend Jürgen. Okay, so I'm not going to spend much time on the actual protocol because we send our tissues to Jürgen and he does it. But the basic premise of the protocol is here. There's a reduction step and a hydrolysis and hydrochloric acid, and then a removal of some of the non-cross-linked amino acids, and then basically put it into a 3 buffer system that's processed in an amino acid analyzer, chromatography system. And an example plot is at the right. This is not for our data. This is just from a review paper talking about this technique, but that's kind of the general idea of the crosslinks analysis. Okay, that's the end of the detour. Back on track. So we did this amongst these other things, and I'll just highlight a few things I'll talk about. We have not looked at the mechanical impact of the anterior capsule. So this is in red because I have not done it. I will talk about looking at hypertrophic changes in the LCL. And then we've done most of this work with the exception of, we haven't yet looked at types of collagen or elastin, which elastin is another love of mine that we've been looking at a lot in tendon, but we haven't yet looked at that here. But everything else in the box I'll talk about. Okay, so what are the data? So we looked at this at these different time points I mentioned, and this schematic up at the left, I'll have on all these next slides just to remind you the groups. So the control would be the white and the gray bars in the data. This is just the time matched controls. So these are just rats that are in their cage, nothing happening to them. The injured ones would be the blue, which is at the end of the immobilization time period. The green is following an additional free mobilization or cage activity period. Okay, so that's all the data is going to look like this. So what did we find? Oh, and then we did 2-way ANOVAs, excuse me, and looked at the significant effects of injury and time, and then in interaction, of course. So I'll just highlight the significant effects. So if we look at total collagen, we see, and this is in the capsule, I'm going to talk about LCL in just a minute. So this is in the capsule tissue. We saw a significant effect with injury. So injured capsules have significantly more collagen. In panel B on the right, after we normalize it by wet weight, there's no effect there. So the density of the collagen is not different, but the total quantity of collagen is increased. When we looked at sulfated GAGs, which, of course, are water binding molecules that are part of proteoglycans, we saw a nearly significant, I'd say, close to significant, but not quite, in terms of sulfated GAGs in the injured capsules, and no change again when we normalize by wet weight. So some subtle changes here in the capsule, increased collagen, slightly increased GAGs following injury. In terms of the crosslinks, so again, we looked at these 4 different crosslinks, 2 immature, 2 mature, and I'll kind of step you through the results here. I know there's a bunch here, but we'll just go through it. So we saw an increase from injury, we saw an increase in this one immature crosslink, DHL-NL, but no change in the other one, HL-NL. And then we saw a decrease in one of the mature crosslinks, LP, lysopyridinoline, but no change in HP. So 1 up, immature, 1 down, mature, due to injury. And then now if we look at significant effects due to time, so from that 42-day time point to 84, we saw a decreased effect for both of the immature crosslinks. So with time, there's less of the immature crosslink, And for both of the mature crosslinks, we saw an increase over time. Now, if we plot these, combine these data together and look at a ratio of immature to mature, we also saw a significant effect with time. So from the 42 to 84 day time point, we see a decrease in the ratio of immature and mature crosslink. So a shift from immature to mature over time. So for the capsule tissue, we saw increased collagen, slightly increased GAGs. We saw increased immature crosslinks and decreased mature crosslinks. And then over time, we see a shift from immature to mature. Okay, with the ligament, we first started with trying to get a better sense of what was happening to the tissue itself. So we did some contrast enhanced micro CT and then segmented out. So I should say these were limbs that we dissected down everything except for the medial and the lateral ligaments. And then we just are imaging the lateral sides, the lateral collateral ligament. And then, like I said, it was contrast enhanced, and then we did a segmentation through the slices of the ligament. So you can see here, this is the control. 42 FM is the control, and 42 IM is the injured. And you can see an increase there in the amount of volume of tissue. And if we look at the 84-day, the control is about the same as the 42-day control, but an increase with the injured one. So you can see that the injury effect is significant. So we see a large increase in the volume of tissue due to injury and no effect due to time. So then we wanted to say mechanically, what effect does that have on this lateral collateral ligament complex? So we reconfigured a previous setup we had to test the limbs and made an adjustment so we could test this now in a uniaxial tensile test for the LCL. Now, I will just say this is not, we had a tough time deciding the best way to do this kind of testing. If you look at the middle picture, we've outlined the edges of the ligament, and it kind of goes in an angled orientation, which you can also see in the schematic in that purple. And we thought about trying to load that along that direction, but then the scarred ones, it's a little hard to say the actual direction. So we decided to load these at 90 degrees and just pull the humeral fixture and just load this in that way until failure. Again, there's maybe some limitations there, but we thought this was the best way to do it. So when we did that, we were somewhat surprised to see there wasn't much different in the maximum force measured at these different time points in between injured and control. There was a significant effect of time, slightly significant with an increase in force with time. But as we thought more about that, we thought, well, it would be nice to get material parameters here. The cross-sectional area is not exactly obvious for this kind of ligament. It's actually more of a triangular ligament with multiple attachment points. And so rather than trying to make a crude estimation of a cross-sectional area, we decided to do a normalized force measurement where we normalize by the measured volume. So not a really traditional way of doing this, but we thought it made sense given the context of the LCL. So if you normalize the max force by the volume of the LCL, then we see these really dramatic differences where there's a really large decrease in the amount of force supported by per unit of tissue, let's say. So although we saw this hypertrophy and a lot more tissue there, it's much weaker quality tissue as we saw this dramatic decrease in mechanics. And then I'll come back to this point later. You may be thinking, well, if you thought that this hypertrophic tissue was restricting motion, then why are the mechanics much less? And what we think is really going on is that this test that we're measuring here, like I said, is pulling in a single orientation. And so we see a decreased force there, normalized force there. But really that massive tissue on the joint, if you think about multi-axial loading the joint and restricting the overall motion of the joint, we think it could certainly still be doing that, even though in this uniaxial test, the force is lower. And then looking at biochemical data, so collagen here, we see a dramatic increase in the amount of collagen or total collagen here, although on the right, there's no change in collagen density. And then we saw really dramatic increases with injury for both the GAGs and even the GAGs normalized by wet weight. So a dramatic increase in GAGs in the injured limbs. With time, we still see an increase in GAGs, no change with time in the collagen. But we do see overall a much more dramatic effect in the biochemical changes in the LCL than we saw in the capsule. I'll point out also there are significant interactions here, which of course complicate the analysis and interpretation a little bit. But I think the trends we're seeing are pretty clear with those two significant effects. And for the crosslinks, we saw an increase in both immature crosslinks in the LCL and this ligament. We saw a decrease in both of the mature crosslinks with injury. And then for time, similar to what we saw with the capsule, we saw a decrease over time of the immature crosslinks, although there was one significant interaction here. And we saw an increase in the mature crosslink hydroxylysyl pyridinoline. So again, looking at that ratio of immature to mature crosslinks, we saw an increase due to injury in this ratio, so shifting towards immature crosslinks, that then over time decreases as there's a maturation process going on. Oh, and there was a significant interaction here too, I should mention. So to summarize the LCL, we saw this vast increase in tissue volume, yet it's a weaker tissue, so poorer quality. We saw increased collagen and GAGs and immature crosslinks, decreased mature crosslinks. Those are all effects due to injury. And then over time, we did still see increased GAGs, and we saw a shift from the immature to the mature crosslinks. Okay, I'm throwing in one other piece of data that's from a subsequent study, but I just thought it's worth mentioning in the context of this discussion. We looked a little more carefully at some of our micro CT, and we noticed this piece of bony-like tissue that we hadn't noticed previously. That's highlighted in red here, that's kind of floating out in the vicinity of the LCL. And it's not exactly clear to us yet where it sits exactly, because these micro CT images were not contrast enhanced to highlight the soft tissues. This is just a standard CT looking at calcified tissues. But we noticed that looking at a similar set of scans for injured animals, we saw that same large piece that was now actually larger and slightly shifted in the anatomical location. And then we also saw a number of other small calcified tissues that we've been calling ectopic calcifications. So if you look at that for the injured, and here I've got both male and female animals, which is what we looked at in this follow-up study. But we see an increase in the injured case, which are all the hash bars. We see an increase in the number of calcifications, the total volume of these, and the total bone volume within these calcified objects. So with injury, we see, as I just said, we see this increase in these little nubs of calcified and ectopic calcified tissue, but really more specific localization and further evaluations needed to determine how these might be contributing. Okay, so to try and summarize some of this and maybe add a few comments, we've shown through this that the anterior capsule and lateral collar ligament hypertrophy are certainly contributing to joint deficiency. And then, as I just showed, we saw also some ectopic calcification that might be contributing to that as well, although we need to look into that in more detail. So what do we see going on? So we saw increased matrix in both the capsule and LCL that we think is leading to a physical barrier that limits joint motion. We did not look at capsule mechanics directly, and it's partly because of the small size and the irregular geometry. But for the LCL, we saw decreased normalized mechanics representing a poorer quality tissue. And then I mentioned this other point that while it didn't cause larger forces along the testing direction for those ligament tests, we think that tissue bulk is restricting multi-axial loading. And then we have a hunch that the deposit of matrix is likely disorganized. We need to do additional studies to confirm that. And we've begun some of that work using some polarized light analysis of some of the sections through the joint. So we saw larger changes in the LCL than the capsule, which was interesting to us, but it does confirm our previous data where we did our sequential dissection showing that over time, that more of the loss of motion is due to the LCL than the capsule tissue. In terms of the crosslinks, to our knowledge, this is the first investigation of crosslinks in a traumatic contracture model. All previous work that we've been able to find was done just in the mobilization only model, which has some subtle differences to it. And this initial increase in immature crosslink indicates new collagen deposition and synthesis, which then over time, as it shifts from immature to mature, we think that's indicative of maturation of the deposited collagen and maturation of this fibrotic scar tissue. So we have a number of unanswered questions. I put a few of them here, but what are the mechanics of this hypertrophic capsule? Is there another technique to evaluate it? Perhaps AFM or some other indentation technique? We haven't done that yet. What kinds of collagen are in the capsule or the ligament, and how are those changing with injury over time? And then I mentioned other compositional changes. We'd love to look at elastin and a potential contribution there. We only looked at a few of the crosslinks. Of course, there's all those other ones that would be challenging to look at, but it's a possibility. And then what is the contribution of those ectopic calcification? In terms of treatment opportunities, I won't have enough time to really go into this in detail, but there are approaches that are targeting a limit of the matrix production. There's a number of different ways to do that, some that we are looking at currently and others. You can limit crosslink formation. This is a little more challenging because of targeting these drugs to the particular tissues of interest and not wanting to restrict normal collagen turnover deposition in other tissues. And then a key takeaway that I've taken from this is that most contracture discussion in the knee and in the elbow really focus on capsule. And we've seen in this and other studies that the ligament really has a large contribution, so we should not ignore it. And then just a quick slide of some ongoing things that we're doing. We're looking at a number of interventional treatment approaches, physical therapy, drug-based therapy, cell-based therapy. We've published on a few of these and others are still in the works. We're also looking, along with Matt Bursey, we're starting to look at inflammation in more detail and looking at the time course of immune cell presence in the capsule tissue first, and then hopefully in the ligament as well. And we got a little side pilot project to look at machine learning algorithm development, to look at automating our digital histopathology and immunohistochemical stains, looking at clustering analysis to look at different cell populations and spatial temporal patterns in the joint. And then we're really excited about trying to correlate some of our histological images with non-invasive imaging like MR to try and look at using non-invasive imaging as a way to understand what's happening biologically in the joint. So with that, I just want to thank my research group. The names in bold over on the left are people who contribute directly to the work I've shown today. And of course, I want to thank our funding and collaborators, and I'm happy to take any other questions.
All right, thanks, Spence. That was great. A lot of information, very comprehensive story, introducing the model and tell us all of these things that you've thought about, which is great. Are there any questions for Spence? I see Adrian has his hand up.
Yeah, that was awesome. I really like all this contracture stuff. I have a question regarding what you were talking about for the ligament at the end. And you measured the maximum force. And I was just curious if there's a way of getting sort of stiffness out of those. Because what if they are much weaker when you break them, but are they stiffer before you break them, or do you have any idea of kind of slope rather than max?
Yeah, that's a great question. And I should have included it. It's in the paper actually. So the stiffness, we did measure stiffness, and actually it follows the exact same pattern. So there was no real difference in stiffness just for the loaded samples. When we normalize stiffness by the volume to kind of get at a modulus type parameter, then it showed the exact same thing. So we saw a dramatic decrease in stiffness for the injured tissues.
Cool, thank you.
Can you remind us, what was the time frame from the start of this project to where you're at? You showed some of the papers when they got accepted, but I always think it's super helpful for junior PIs like myself, but also students and postdocs to kind of get a sense for, they see this fantastic work, and you shared with us some of the deviations, but we forget how long these things take. So I'm just curious, sort of from the conception to where you are today, what time frame are we talking about?
Yeah, that's a great question. And for this project, it really has been a long time coming. I first talked about this idea when I interviewed at WashU, which was spring of 2012, so 10 years ago. And we started work on it really kind of start of 2013. And we got our first funding. We had a small pilot grant from the American Shoulder and Elbow Society, and then an RL3 in probably 2014 or something. But our first paper wasn't published till 2016. So from idea of the project, conception of the project till our first paper was 4 years. And then in the subsequent 6 years, we've done a lot of development here in a number of these different areas. But yeah, to that point, it's very true. It took a number of years to kind of get the model up and going and validated. And then we're excited for all the things we can do with it now. And I didn't talk about a lot of stuff we're doing. I had a PhD student finish last year, did a lot of nice work looking at functional assays. So looking at gait analysis of these rats and tracking forelimb function and a number of other things that I didn't have time to touch on. But yeah, it's been a lot of fun seeing the project grow and expand over time.
Any other questions? don't be afraid to just unmute or in the chat, one way or the other.
Hi,
Colleen.
can you hear me? Okay. So I'm really excited about the cross-linking work that you talked about, because we have some projects where we're trying to sort of measure and think about cross-linking. And so I guess my question to you is, do you think that the cross-linking assays and techniques that you developed are more universal for cardiovascular tissues, since of course that's what I care about, or do you think that they're really specific to like tendons and orthopedic tissues?
No, they're not specific. Yeah, they're not specific. So they would be fine to be used for you know, other tissues. The digestion protocols might be shifted a little bit, you know, to get the tissues fully digested. But you know, if it works for tendon, which is a pretty dense collagenous tissue, it should probably work for other types of tissues. But yeah, that could be used for these other ones as well.
And then do you have any suggestions on finding collaborators who can implement these things? Because clearly that was a struggle.
Yeah, you know, I showed that slide just to show that, you know, sometimes it's not the easiest thing to find. Well, you could certainly ask around your own institution, because, you know, people that have chromatography setups, you know, might be able to do that. For the protocols we use, there's a couple of requirements. Like I said, you have to be able to measure fluorescence to identify some of the crosslinks, but certainly start at your own institution, and then, you know, I'll be happy to talk to you offline about, you know, connecting you with our collaborator. He's not going to want me to send everybody there probably, but connecting with him. And then, like I said, there are people around that are interested in perhaps doing this. I mentioned Catherine Poe. She might be up and doing it now in Maryland, where she's now, I'm not sure. But you know, just start asking around. I think you can probably find people that can help.
Awesome, thanks.
Seems like Jürgen may be getting 50, 100 emails after this. I don't know.
Yeah, I hesitated to highlight his name, but but he really was a lifesaver, and he's been fantastic to work with. So he's been a great collaborator.
I had a quick question. I think the cross-linking stuff is super interesting because, you know, people say, oh, cross-linking could change, but here you've actually measured it, right? And I think it's nice to actually see that. But in the LCLs, I guess, were you able to look at orientation of collagen somehow? I mean, is it more aligned along the main axis or perpendicular to the main axis of the ligament? Is there a way to do that?
Yeah, that's a great question. I wish I had that data. We haven't looked at that yet. We definitely need to do that. My hunch, just by looking at the dissections of these, is that this collagen appears just to be kind of thrown down, and I'm guessing that it's not aligned much at all. The ligament, the healthy ligament, like I said, it's somewhat triangular in humans as well. And we've done separate studies of this ligament in humans, but it is more triangular. So you do have collagen. It's not like a typical ligament or tendon that it's all aligned in one direction. You do have a bit of variation in that triangular shape. But having said that, it's still well defined and organized. I think in the injured case, I think it's much more disorganized, and we do have ways we can look at that. You know, we do a lot of polarization imaging of, you know, sections, or if relatively thin tissues, we could do that. We just haven't done that yet, but it would really be really interesting, and I think it would show a lot about what we saw with the mechanics if we could have a better description of how the collagen is organized.
If there's no other questions, I have one more question about the cross-linking. And this may be a naive question, and I know that you premised by saying you're not a biochemist, so you maybe don't need to have the exact answer, but I'm curious about how, you know, the whole process basically of cross-linking, like how much is that a cell-mediated process versus purely a biochemical enzymatic process? Like if you just had collagen there, will it cross-link and mature in the same way, or do you need cells to do that?
That's a really good question. So I know that cells will modulate the amount of lysyl oxidase, you know, the enzyme that's critical for that. And there are, you know, there's a whole family of lysyl oxidase proteins. There's LOX itself, and there's lysyl oxidase-like proteins, and some of those are more common in other tissues than in different tissues. So we know from some work in tendon that lysyl oxidase-like 2 is the most common of those. And so you have to have those enzymes to be able to do this enzymatic cross-link. So that would need to be present. But beyond that aspect of it, in terms of the cells producing the enzyme, I don't know how much they're directly involved in the cross-linking. It seems to me that it's more of a biochemical process outside the cell after the collagen molecules have been produced and secreted. But again, I will hesitate on the final details since I'm not an expert in the biochemical processes here.
Yeah, I think that makes total sense. I mean, you need the enzymes there. The cells are probably going to be producing them. But maybe to Colleen's point, there may be some different levels of lysyl oxidase expression that could modulate cross-linking in different tissues.
Yeah, absolutely. And I think that is well established. I don't know that would change the measurement of the cross-links, but in terms of the amount of cross-linking and which enzymes are actually causing those to occur, yeah, I think that does vary by tissue.
Maybe I'll ask another question. Is this process at all similar to wound healing I guess I'm interested in wound healing in skin, but is there some sort of sequence of inflammation cascades that triggers the whole hypertrophy cascade?
Yeah, very good question, Adrian. So yes, we think there's some similarities to typical kind of wound healing or contracture of a wound, even in skin, for example. I mentioned we're just starting to look at inflammation. We've looked in general terms at histology. At the time points I showed mostly here today, we saw very little inflammation, but those were the kind of the longest time points from any sort of actual impact to the joint. At our intermediate time points that I showed none of this today, but intermediate time points, we have seen some influx of an inflammatory response. We're currently looking at that together with Matt, looking at capsules at different time points and looking at the immune cells that are present in the capsule at least. So we should have some idea of that. But the general kind of process that you alluded to is that we think there is an inflammatory response. We also know that there is myofibroblasts or conversion of fibroblasts to myofibroblasts that will be more contractile, right? Will contract the tissue. And some of that's been shown in clinical samples from contracted elbow joints and in a number of kind of rabbit knee models, which is a common model to look at contracture. So yeah, there's a number of things going on here, and some of them absolutely share similarities to other wound healing responses, like in the skin.
Thank you.
Okay, in the last few minutes, any burning questions? Last thoughts for the speaker?
Just want to reflect what people said. That was a really nice story arc. I appreciated that a lot.
Well, thank you. This was actually, I'll just say, this was a really fun format to be invited to present because, I knew what the paper was, but I went back and really dug deep into it and thought, why did we do this? And what does it really mean? And how does it fit? And it gave me a chance to really dig deeply into this one paper of many on this project and really think about it. And so it was a fun exercise. So yeah, I appreciate the invitation.
Yeah, I definitely want to echo what Manuel said. I actually saw one of your students present a subset of this work at SP3C, I think one of the virtual meetings, and it was just super nice to sort of see the overview of the project, because I remember being really impressed with the work at the time, and it's good to hear that it's like a decade-long project and see all the pieces.
Yeah, well, thanks, I appreciate that.
Okay. Maybe with that, we can stop the recording, end the session for now. Thanks again, Spence, and we'll see everyone in 2 weeks for the next Journal Club.